All right, sounds good. All right, hello everyone. My name is David Bianchi, and I'm a graduate student in the Luthi Schulten group at UIUC, who's been working with the synthetic biology group at JCVI for the last couple years, mostly in developing computational models and some data analysis as well. So working with Andras a bit on the last project, but also what I'm going to focus on today is trying to build a dynamic model for cell growth in Syn3A. And so what really inspired us looking at Syn3A in general, kind of touching on Andras and John's talks, is this idea that the NSF put out a couple years ago, understanding the rules of life. And so they have this really cool logo here over on the left, but a few specific subpoints that we were quite interested in is, can we predict the phenotype of Syn3A from some basic rules or characteristics that we can see? So something really interesting for computational modelers to try to think about. Can we make some connection between the energy usage of the cell and how this is related to how its genes get expressed and how its metabolism produces this energy? So something that Andres really talked about in the last talk. And then finally, can we understand this in a multi-scale type of way, where we're spanning multiple time scales, all the way from the time scales of smaller sub-processes in the cell, like DNA replication, to multiple cell cycles, and across different levels of biological organization in the cell as well. And so the kind of overall goal we came up with, which one of my colleagues, Zane, is going to be talking about more later today, is making this multi-scale kinetic model for JCVI Syn3A. And how this really connects to that overall goal of understanding the rules of life is what I would say would be through a few sub-goals. One of which is having individualized gene expression for all of the genes in Syn3A, and linking this then to mRNA and protein expression, since we only have less than 500 genes now due to the genome reduction that the JCVI has undertaken. That we have a multi-scale model, like I mentioned before, so we can understand how these processes are really interconnected. That we have modular cell processes, for example, for how the DNA of the cell gets replicated, or for how the cell grows, that I'm going to talk about today, and to really understand how these are intricately connected to gene expression and metabolism. And then finally, a model that integrates experiment and theory. And so what I'll really focus on today is this modular cell process for cell growth that we came up with. And so as Andres mentioned, the really key starting point here for us was some work done by Professor Marion Breuer in developing this metabolic map for Syn3A that we use for our FBA model. And so something that's really key to note, I think, both from just looking at the map and from Andres's talk, is that all of these networks, in terms of producing lipids, the main glycolysis pathway, are very interconnected and have this kind of communication and exchange through various metabolites that are in the system. And so even just looking at this small little patch of producing glycolipids here that I have on the left, we can see that this system's taking in glucose 6-phosphate from the central metabolism and UTP from the nucleotide metabolism to make this eventual glycolipid that we're going to see in the membrane. And so this kind of idea of link metabolites that are linking and communicate between these different subnetworks is going to be really crucial for us. Another idea that turns out to be really important, I think, just in cellular biology overall, is that the concentrations that you see in a cell span a really massive range. And so we can have something like the cellular DNA or RNA that might only be present in 1 to 10 copies, perhaps, in the cell, really sparsely concentrated. And then you can have things all the way on the other end of the scale, like ATP and amino acids that we're going to use in translating proteins that might be present in millions of particles and millimolar concentrations. And so what's really becomes apparent is that we have to model these processes and these chemicals in different ways. And so in our multi-scale model, we choose to do this by modeling the metabolism and the gene expression a little bit differently, but trying to link these two processes together so we can really understand that interplay. And so eventually we come up with a model that has over 200 metabolites, has expression of every gene of protein, and has enzymes for this metabolism who their number is expressed and updated in these metabolic flux solutions. And so we do this by this hybrid method that we developed a few years ago and tested computationally, where we simulate for a given time step here of 1 second, stochastic chemical master equation, where we can see these gene expression reactions, like for example, transcription of a gene to RNA or translation of a gene to protein. And then after this time step, we'll then update our metabolic simulations, which we just do by simple ODEs with these new enzyme counts, and then simulate this metabolism out for that time step. And what this is going to then update in our gene expression reactions is, for example, the amount of NTPs that you might have available for transcription or dNTPs that you have available to replicate the DNA. And so with all these tools and kind of principles in place, what we can then do is get to our eventual goal of building this cell growth module. And so this is some work that we did with the group of Professor James Sands at the TU Dresden and his graduate student, Natalia, who's going to be giving a talk next Monday. And so the lipid pathway of Syn3A is relatively simple. We can take up fatty acids and glycerol from this FBS or Albumax knockout-based medium that many of you might be using, provided by Kim Wise, and we'll eventually use these products to produce lipids like phospholipids or glycolipids, like I mentioned before, as well as just taking up certain lipids directly. And then, as I mentioned, we could also produce these glycolipids that I touched on earlier. We had previously thought that we might produce a lipid-lactam capsule as well, but this, through some work with collaborators at UAB through GCMS, this was shown not to be present in Syn3A. And so the really interesting experimental input that helped us really refine this model was some lipidomics work done by the Sands lab, where they were able to identify that Syn3A has very cholesterol-heavy membrane, and what was a little bit more surprising to us, also fairly heavy in sphingomyelin that it's able to take up directly, as well as phosphatidylcholine. And so with this data, we were able to then update our lipid biomass from the FBA and really try to quantify more the amount of phospholipids, sphingolipids, and neutrolipids relative to each other here in the lipidome. And so with this eventual kind of production goal for our cellular economy in place, we're then able to build up this model. And it's important to keep in mind that in making our membrane and having our cell grow, it's not only going to be lipids that contribute to the cell surface area, but also proteins that get translocated into the membrane. And I think this image by David Goodsell really encapsulates that quite well. And so in our model, what we're able to do then is take measured lipid head group surface areas from monolayer experiments that James Sands conducted, as well as from some MD simulations, and use these to continue growing the surface area of the cell as lipids are produced by our metabolism or taken up, and as different membrane proteins are translated by our gene expression module and then translocated by the sex system into the membrane. And so what this model then allows us to predict over a population of hundreds of individual cells with stochastic gene expression is doubling times for Syn3A. And so via our simulations here, we can see a range of doubling times on the order of 90 minutes to 110 minutes for Syn3A, with an average just under 100 minutes. And we can see kind of in the shading here, interquartile range of these hundreds of cells for the doubling time, centered about 100 minutes. And then the individual contributions from membrane proteins in blue and lipids in red. And the final really interesting thing that we can note is the noise in all of these different components from cell to cell increases with cell cycle time as we get stochastic expression of both membrane proteins and some key lipid metabolism enzymes that are going to make our final phospholipid products, for example. And so in
Okay.
conclusion, now I'm running out of time here, John. We've developed a model with the following features: a multi-scale model for cell growth that's determined by and responsive to both the metabolism and the gene expression of Synth3A. We integrate experiments, lipidomics done by the SINS group, as well as proteomics and theory to predict the phenotype of doubling time for these cells, as well as some key metabolic choke points in the lipid metabolism that I didn't get to talk about today, but that'll be in our paper that's currently under review. And then we can capture cell-to-cell variation, which my colleagues will talk about a little bit more in their talks later today. And then in future work, we hope to develop a spatial growth model that inserts membrane proteins at specific locations in the cell as that volume of the cell is dynamically expanding. My colleague Maddie will be speaking about that later today. And then on the experimental side, studying how that membrane composition changes in response to introduction of a new medium or temperature changes. And Natalia and Isaac from the SANS lab will be talking about that next Monday. So thank you so much all for your attention. I'd be happy to take any questions at this point. And thank you so much for the SANS lab, especially Professor Kim Wise, my advisor, Professor Lee Schulten, and Zane, who worked on me with the computational model for being such excellent collaborators. Thanks again.
Okay, nice talk, David. Questions?
Quick one.
Please
Hi,
identify
Dave.
yourself.
Elibio, Rich. Dave, nice talk. I'd like your comment relating interactive machine learning approach, maybe directed what Andy, the first talk just mentioned about ATP, would be useful to try some experiments to accelerate and de-accelerate the growth in order to check whether interactive machine learning techniques would be useful for the whole process.
Yeah, I think that's a very interesting idea and something we've discussed in the group and played around with a little bit and hope to work on in the future. And I think there's even a few other groups, John can correct me if I'm wrong, who are thinking along the same lines that we've spoken with a bit before. So that's definitely something that we've already begun working on and studied a little bit in our work that's going to be published soon and that we want to go further with into the future here.
Thank you, Dave.
Thank you, Avery.
Anyone else?